Hello to everyone, I'm Chamihan from Malaysia, and today I'm going to present to you a part of my PhD project entitled Antibiogram and Molecular Typing of Multi-Drug Resistant Staphylococcus aureus isolated for veterinarians and livestock farmers. Before I proceed further, I would like to briefly introduce myself and where I'm from. To those of you who are unfamiliar with Malaysia, Malaysia is a country located in the southeast of Asia, consists of 13 different states and 3 federal territories, mostly in the peninsula and other 2 in the Borneo Island. UNISA, where I'm currently pursuing my PhD, is a public university located near the east coast states of Terengganu, and Malaysia is a multicultural and multi-ethnic country, and I hope that one day you will have the opportunity to visit my country soon. So let's back to my presentation for today. For introductions, antimicrobial resistance, or AMR in short, is a global concern as new resistant mechanisms are emerging and spreading globally, threatening our ability to treat common infectious diseases, resulting in prolonged illnesses, disability, and death in both human and animal. And taking Staphylococcus aureus as example, Staph aureus is known to be an important pathogen in both humans and livestock animals. Today, we have noticed that many different strands of Staph aureus have developed resistance towards various commonly used antibiotics. In some cases, some of these Staph aureus display multi-drug resistance or MDR patterns, such as the famous medicine-resistant Staph aureus (MDRSA). They were first identified from clinical specimens in UK in 1961, and nowadays MDR have become an emerging threat worldwide in both clinical and veterinary practices. Previous studies in Europe, the United States, and elsewhere have indicated that individuals with constant exposure to animals, such as animal farmers, are at risk in contact with AMR bacteria. In Malaysia, previous studies have shown that the rise of MDRSA occurrence rate from 18% in 2016 to 19.8% in 2017. However, data regarding the phenotypic and genotypic characteristics of MDRSA isolates from animal handlers in Malaysia is rather limited, and thus our teams have carried out projects with the following objective. Number 1, we try to determine the phenotypic antimicrobial resistance profile of the MDRSA isolates from animal handlers, and second, we will categorize the isolates using SPA, AGR, and ISC typing. And in order to fulfill the objective of these studies, we have selected 30 non-repetitive Staph aureus isolates. They were previously isolated from 398 animal handlers in various states in Malaysia. The 30 isolates were confirmed to display phenotypic multi-drug resistant patterns using capable methods. Then, PCR amplifications of IEC, SPA, and AGR genes were carried out, followed by DNA sequencing of SPA genes to determine the SPA genotypes. In addition, we ran some cluster and diversity index analysis to determine the possible relationship of the isolates. And now, let us have a look at the result and discussions. In regards to the phenotypic antimicrobial resistant patterns, we found that more than 90% of the tested isolates show resistance against 4 and above type of antibiotics. These isolates have a MAR index of 0.22 and above, which is higher than the 0.2 threshold value for the multiple antimicrobial resistance index assessments, suggesting that these isolates may originate from environment with high antibiotic usage or contaminations. Majority of isolates were resistant to antibiotics from beta-lactams, microlids, and tetracycline groups. And besides, based on the bar on the right, we can see that the majority of isolates show the highest resistance rate against penicillins, followed by amoxicillins, erythromycin, and tetracycline. However, no resistance was observed against gentamicin and linezolid. Recently, information regarding antibiotic resistant profile and molecular characteristics of the Staph aureus have changed rapidly around the globe, and thus continuous surveillance of antibiotic-resistant Staph aureus, especially those with MDR trait in high-risk individuals such as animal handlers, is highly recommended to obtain the latest data regarding the antibiogram patterns as well as the genotypic characteristics of the bacteria. In these studies, the isolates were highly resistant against different antibiotics, especially those from beta-lactams, tetracycline, and microlid groups. These findings were higher than the antibiotic resistance profile reported by the study in Switzerland, where Staph aureus from pig farmers and veterinarians show relatively lower resistance rate against penicillin, tetracycline, and erythromycins. A more recent study in 2017 reported that MDRSA isolated from pigs and poultry farmers show relatively higher resistance rate against penicillin, tetracycline, and erythromycin. Despite the high resistance rate against certain antibiotics, all of the isolants were sensitive against linezolid and gentamicin, suggesting that these two antibiotics are viable to treat Staph aureus infections among animal handlers in Malaysia community. However, as gentamicin and linezolid are considered to be a critical importance antimicrobial agents by WHO, the usage of these two antibiotics should be carefully monitored and well regulated. Meanwhile, for ISC genes, we detected the presence of 3 genes that were previously associated with human immune evasions, including SAK, SCN, and CSP genes. Further characterizations of MDRSA genotype based on their combination of ISC genes revealed ISC type E was the most commonly found ISC types. In addition, only 2 AGR groups were detected, with AGR group 1 being the prominent AGR subgroups. In these studies, we applied the use of PCR to determine the prevalent ISC so that we can determine the origin source of the Staph aureus. In more cases, ISC genes, particularly SN genes, were not frequently detected in Staph aureus originated from animal, and thus, the absence of such genes in Staph aureus will be considered to be a marker and strong indications for the livestock-derived strands. In these studies, SN genes was not detected in 76.7% of the MDRSA isolants. Previous studies have demonstrated that isolants from humans with occupational exposure to animals may not harbor ISC genes, possibly due to transmissions and colonizations of Staph aureus from animals, and thus the disappearance of ISC genes in Staph aureus may possibly be due to the genome alterations, including the loss of genetic elements that carry ISC genes during the animal adaptation processes. In these studies, ISC variant type E was the most frequently found ISC types. This finding is different to previous studies that reported that high prevalence of ISC positive S. aureus for clinical sample obtained from hospital patients and persistent Staph aureus carrier with the predominant ISC type being type B. The findings suggest that there may be a variations of ISC type according to sample origin as well geographical regions. Using conventional PCR sequencing analysis for the 30 MDRSA isolants showed 70 different SPA types, where type T189 and T2174 were the most commonly found genotype, followed by T4171 as shown in the table. The phylogenetic tree displayed on the right was developed based on the DNA sequencing of the SPA genes. The phylogenetic tree shows that the SPA type can be divided into 2 major clans, as shown in the green and red line boxes. Aside from the SPA genes phylogenetic tree, we are also interested to know whether the isolants are related based on their phenotypic and genotypic trait. So we constructed a dendrogram using the phenotypic and genotypic data that I mentioned in the previous slide before. This dendrogram showed 2 major clusters were formed. Subsequently, 16 smaller clusters were detected, where 70% similarity was used as a threshold to cluster the isolants. And based on the dendrogram, we can see that the isolants seems to be diverse from each other. And to further confirm these findings, we also run Simpson Index and Simpson Index of Diversity analysis to determine the diversity of the isolants. And based on the result displayed on this table, we can see that the isolants are greatly diverse from each other based on their phenotypic resistance patterns, SPA type, and combination of both phenotypic as well as genotypic traits. SPA typing of MDRSA shows the presence of 17 different genotypes, where showing that there is a great diversity of SPA type among the isolated MDRSA. In the current study, the most commonly found genotype are SPA type T189 and T4171. And for information, the presence of these 2 genotypes were previously reported among Staph aureus isolated from hospital patients, university students, and poultry farm workers in Malaysia. In the present study, AGR type 1 was the most commonly found AGR subgroup, followed by AGR 2. Similar findings were also reported in Korea and Malaysia, where they reported that AGR type 1 being the most commonly found type among the Staph aureus, with the prevalence rate ranging from 64% to 100%. In conclusions, this will have managed to detect and determine the presence of MDRSA isolants with different phenotypic resistance patterns, ISC types, AGR group, as well as SPA genotypes. The findings suggest the emergence of MDRSA assurance with great genetic diversities among animal handlers in Malaysia, and thus may possess public health threat in the future. As staph infection have proven to cause genetic implications, transmission of these pathogens between animals and animal handlers is strongly suspected, and thus we will recommend a more comprehensive surveillance and investigations conducted on staph and MDRSA isolated from both humans and animals. And these are some of the references that I use. Before I end my presentations, I would like to express my gratitude to IIGS for funding these studies. Furthermore, special thanks to all the undergraduate and postgraduate students of UNISA, the staff of the Department of Veterinary Services of Terengganu, as well as University Kelantan, Malaysia for the help during the sampling and data collection processes. Finally, special thanks to the event committee for giving me the chance to present here. And with that, thank you all for listening.